Hello boys, welcome back to another interesting video tutorials, and this is meant for class 12th biology students, and this is for 7th September 2020. Hoping that all of you have done very well in your UPSC ND exam yesterday, with the hope that all of you would become defense officers and wearing uniform sooner or later. We go with the overview of what we have been studying in this chapter number 11, Biotechnology, Principles and Processes. We started off with the very definition of biotechnology. Biotechnology is the use of living organisms in systems or processes for the manufacture of useful products, we said. So it involves normally algae, bacteria, fungi, yeast and cells of higher plants and animals. So basically this is for isolation of DNA, and the DNA must be having gene of interest. So we require some tools for that. If you recollect from your memory, we said tools are first and foremost is enzymes, in that cleaving enzymes, exonucleases, endonucleases, restriction endonucleases, lysing enzymes, synthesizing enzymes, ligases, and alkaline phosphatases. These are all the various enzymes we listed that we require. And then we talked about step number 2, that is the separation and isolation of DNA fragments through gel electrophoresis. And then in our last session we talked about cloning vectors, and in that particularly we discussed about PBR322 vector, an first artificial cloning vector constructed by two great scientists, Bolivar and Rodriguez. That's why the name PBR, P for plasmid, B for Bolivar, R for Rodriguez, and PBR 322. And then we were also talking about the competent host, how it should be done, how the DNA should be inserted in the competent host. And we said electroporation, and then gene gun method or ballistic method, and then micro injection, and then chemical mediated gene transfer like polyethylene glycol, we said. And now in this session we are talking about the processes. Once we got all the tools, now we focus on the processes of rDNA technology. In that processes, the following are the steps. Number 1, isolation of genetic material, that is DNA. Second, fragmentation of DNA by restriction endonucleases. Third, isolation of desired DNA fragments, gene of interest. Fourth, ligation of the DNA fragments into a vector. And then once we obtain rDNA, transferring rDNA into a suitable host, and then culturing host cells in a medium at large scale. So we are going for not only quality, for quantity also. And then what is the aim of all this? We said to obtain desired products. So in the last step, with a very valuable one, extraction of desired product. Now we go with the first step, isolation of genetic material, DNA. So here most of the eukaryotic cells have DNA as their genetic material. And to form rDNA, the DNA should be pure, free from all biomolecules. And the DNA located inside nucleus, enclosed by plasma membrane, and genes intertwined with the histone protein that we said. And cells have to break open to release DNA and other macromolecules such as RNA, proteins, polysaccharides and lipids. So cells are lysed with lytic enzymes like lysozyme in bacteria, cellulase in plant cells so that cellulose will be digested, and chitinase in fungus because chitin will be there. And the macromolecules treated with specific enzymes to remove them, like RNA will be removed by using an enzyme called ribonuclease. Proteins will be removed by using an enzyme called protease. So this is how we eliminate one after the other all the macromolecules, and finally we get DNA. So purified DNA precipitates with the addition of ethanol, and it appears like a collection of fine threads in suspension. And this experiment each one of you will be doing in our lab once you come back to the school, and you will be very happy to do this experiment. So this is how you get that white threads here. So this is how you obtain. The cells are lysed using a detergent that disrupts the plasma membrane. This is the first one. So you are using detergent soap liquid. Cell contents are treated with protease to destroy protein and RNase to destroy RNA. Step number 2. And step number 3, cell debris is pelleted in a centrifugal machine. We have got a machine. We do the centrifugal process and we get a supernatant liquid containing the DNA is transferred to a clean tube. So this is how we get threads. The DNA is precipitated with ethanol. It forms viscous strands that can be spooled on a glass rod. So each one of you will be performing this experiment and extract the DNA. And here you can also see the cutting of DNA at specific locations. So in order to get pure DNA molecules, the following steps can be taken. Bacterial cells are treated with lysozyme to dissolve the bacterial cell walls. Plant cells are treated with enzyme cellulase to dissolve cellulose cell wall. And the enzyme chitinase is used to dissolve fungal cell wall. The plasma membrane degrading enzymes are used to degrade membranes. This results in the release of DNA along with several other macromolecules and impurities such as RNA, proteins, polysaccharides, and lipids. The eukaryotic DNA molecules are intertwined with the proteins such as histones. These proteins can be removed by treatment with an enzyme protease. The enzyme protease converts proteins into amino acids. Similarly, RNAs are removed by treating with an enzyme called ribonuclease. So other macromolecules can also be removed by specific treatments. And finally, pure DNA molecules are precipitated out by adding chilled ethanol and collected as suspension. So once we obtain the DNA, now cutting and ligation of DNA at specific locations. The vector DNA, that is plasmid DNA, and foreign DNA, alien DNA carrying gene of interest are cut by the same restriction endonuclease to produce complementary sticky ends. Remember, we were talking about the sticky ends. This process of cutting DNA by restriction enzyme is called restriction digestion. With the help of DNA ligase enzyme, the complementary sticky ends of two DNAs are joined. This process is called annealing. So annealing to produce rDNA. So this is how we get that the rDNA. So step number 1, purified DNA. We have just now said source DNA, desirable DNA, vector DNA, incubated with specified restriction enzymes for the process. So digestion at optimal condition. Progression of restriction enzyme digestion is checked. Agarose gel electrophoresis. We were talking about that in our previous sections, gel electrophoresis. And DNA being negatively charged, so move towards positive electrode, anode. And once gene of interest, cut vector with the space for gene of interest is isolated and DNA ligase is used to add up. And this step is preparation of rDNA. This is how we prepare rDNA. Now we go to the diagram part of it. You can see here. Yes. Digestion with this. You can see digestion with restriction enzymes. Cleaved DNA. Your cleaved DNA. Then of course electrophoresis. And here plasmid vectors, cleaved vectors. And then electrophoresis to detect progression of restriction enzyme and ligation of gene of interest and vector. So this is how we obtain. We use the gel electrophoresis to purify the gene of interest and we get the vector DNA. Now the third one, most important one. Once we obtain what we wanted, gene of interest, amplification of gene of interest using a method called PCR. What is PCR? Polymerase chain reaction. So PCR is polymerase chain reaction. This technology is used to amplify a single or a few copies of piece of DNA to generate in large quantity, thousands to millions of copies of a particular DNA sequence in vitro. So components required, we need a primer. Primer is a small chemically synthesized oligonucleotides that are complementary to the regions of DNA. So we require primer, then we require nucleotides, and then DNA polymerase, an enzyme. So incubated together, then these are the things that we require in PCR. And what are the steps involved in that? In PCR, there are mainly three steps. One is denaturation, second is annealing, third is polymerization or extension. So denaturation, the target DNA is heated to a high temperature, usually 94 to 98 degrees centigrade, resulting in the separation of two strands. Each single strand of the target DNA then acts as a template for DNA synthesis. This is denaturation. This is what we do. And then primer is added, so which is binds to complementary region. This is called annealing. So two oligonucleotide primers anneal to each of the single stranded template DNA, since the sequence of primers is complementary to 3' ends of the template DNA. This step is carried out at a lower temperature, usually 40 to 60 degrees centigrade, depending on the length and sequence of the primers. And then polymerization, DNA polymerization. So here polymerization with nucleotides in the medium. Genomic DNA act as template. That means tuck DNA polymerase, we say TAQ, tuck. From a thermophilic bacterium, the name of the bacterium you must remember, Thermus aquaticus. I repeat, Thermus aquaticus or aquaticans. So basically we use this Thermus aquaticus. It synthesizes the DNA region between the two primers using DNTPs. What is this DNTPs? D, deoxy, N, nucleoside, T, tri, P, phosphates. So deoxy, nucleoside, triphosphates. And of course, Mg2 plus is a must. It means the primes are extended towards each other so that the DNA segment lying between the two primers is copied. This occurs at an optimum temperature of 72 degrees centigrade. See, various temperatures are there. So you can see here, we can synthesize 1 billion copies of desirable DNA. That's how we are using the word amplification. You can see here, reason to be amplified, that red color one. So step number one, this is the red colored one, this zone, reason to be amplified. And this step is denaturation. And you must remember in denaturation, 94 to 98 degrees centigrade, that much temperature is required. And then the second one is annealing. In annealing, it is something like 40 to 60 degrees centigrade. This is to be maintained in annealing. You can see the primers. So we are extending the primers 3' to 5' and 5' to 3'. And for this, what we are using? The DNA polymerase, tuck polymerase. What is the tuck polymerase? Thermus aquaticus. It is thermophilic bacterium. So tuck DNA polymerase synthesizes the DNA region between the primers using DNTPs and Mg2+. So you must remember here, Mg2+ is a must. Mg2+ and DNTPs. DNTPs. What is the DNTPs? As I said just now, deoxynucleoside triphosphates. So this makes the primers extend towards each other, as you are seeing. See, this one is extending and this is extending. So we are getting and repetition of 30 cycles. So we get 1 billion times. So this is how what we do, we amplify. The PCR, it was originally invented by, you must remember the great gentleman, Carey Mullis in 1985. PCR results in the selective amplification. That is the purpose. What is that we are selecting? The primers. Selective amplification of a specific region of DNA molecule, and so can also be used to generate microgram quantities of DNA copies up to 1 billion of the desired DNA present even as a single copy in initial preparation. A single PCR amplification cycle involves 3 steps: denaturation, annealing, and extension. So this is how we amplify the gene of interest. So in our next session, we'll be talking about insertion of rDNA into suitable host and then obtaining the final product, that is a desirable gene or gene of interest, through some processes like downstream processing, using bioreactors, all that. That will mark the end of this chapter. Until then,